Okay. So hi everyone. Thank you for coming. I think I've met all of you, but if not, my name is Marco. I'm a PhD student working with Dr. Raw, and I'm now co-supervised by Dr. Rob Vanner over at Princess Margaret. And today I'm here to talk about my work over the last year or so, which is trying to understand what clonal hematopoiesis is doing in the tumor microenvironment. So to start with a little bit of background, we study clonal hematopoiesis. It's often abbreviated as CH. Sometimes you'll also hear it called clonal hematopoiesis of indeterminate potential. That stands for CHIP. The terms are used largely interchangeably, and I'll probably use them interchangeably today. But what CHIP is actually describing is a pre-malignant condition in the hematopoietic system or the blood. Um, so as you're going throughout your life, you have tens to hundreds of thousands of hematopoietic stem cells that are continuously growing and proliferating. These are the cells that are required in your body to produce your blood, and immune cells, which are, of course, vital to your survival. Um, and as these cells are growing and proliferating, it's almost inevitable that they're going to acquire mutations in their DNA through replication and cell division. Most of the time, these mutations are essentially harmless and they won't really do anything. But occasionally, these mutations can fall on cancer driver genes. So these are genes that are recurrently mutated in leukemias and other blood cancers. And these are genes that can provide a selective advantage to these hematopoietic stem cells. And by around age 40, these mutations are actually ubiquitous in the population. They're present in just about everyone. But what's really interesting is they only really hang out in one or two cells, and they don't really have any consequences in most people. But as aging continues, and once you reach around age 65, these cells begin to expand. So they begin to take up a larger proportion of your total hematopoietic compartment, and they begin to produce a larger proportion of your blood cells. They begin to skew hematopoiesis, and your downstream immune cells tend to be dominated by this single clone, which is where the name clonal hematopoiesis really comes from. And the implications of this are really vast in human health. Of course, CHIP was initially discovered as a precursor state for hematologic cancers, and it's of course able to continue acquiring driver mutations and become a cancer, like Laura was talking about last week. Um, but what you might know about leukemia is it's a lot rarer in the population than CHIP is, especially a lot rarer than 20% of all elderly folks. And the implication of that is a lot of people are going throughout their life, especially reaching towards the end of their life, and they just have clonal hematopoiesis circulating in their blood, but they don't have any associating blood cancer. Um, and with this, what happens is clonal hematopoiesis is actually able to leverage inflammation to expand and grow in the blood system. It leads to a lot of systemic inflammation in the body, and an extension of that is that systemic inflammation is related to a number of different age-related diseases that I'll circle back to in a little bit. So now looking at the stem cell level and where clonal hematopoiesis is really coming from, I mentioned that in most people, these mutations aren't really given the conditions that they need to grow and expand. But in the context of some sort of inflammatory stimulus, usually this is a stimulus that comes from aging. So as we age, inflammation just tends to accumulate in the body. But there could be other stressors, like, for example, an acute infection that lead to a spurt of inflammation in the body. And what this inflammation does is it allows these initial clones to expand and begin producing more and more mutated blood cells. And what's important is that the immune cells that these mutated cells produce are still carrying the mutations of their initial stem cell clone. And these mutations lead to disrupted immune cell function. There's a number of different changes that happen. One main thing is there's a myeloid skewing of hematopoiesis. So what that means is you get a lot more myeloid cells that are produced. These are cells like macrophages or monocytes, and as well as your granulocytes, which are cells like neutrophils. And what also happens is these mutant cells that are produced are dysfunctional. So they tend to have impaired phagocytic capacities, and they tend to release a lot more pro-inflammatory cytokines. These are cytokines like IL-6 and IL-1 beta, and these are cytokines that feed back into the bone marrow and, I guess, propagate the selective advantage of these hematopoietic stem cells. So the inflammation goes back to the bone marrow and allows these cells to continue growing in sort of a positive feedback loop. And actually, the genetic level of the stem cells in the body, the most common mutations that cause CHIP are DNMT3A and TET2. There are a number of others, but these are, I guess, vastly the most common. They account for about 80% of CHIP mutations. And what these mutations do is they give these stem cells an intrinsic resistance to inflammation. Typically, when your hematopoietic stem cells come into contact with an inflammatory environment, they kind of regress, and they're kind of hiding and just trying to survive the stress, and they're not able to continue proliferating. Whereas the mutated stem cells are actually able to grow and continue proliferating and producing your blood cells, which is what generates this initial advantage and ultimately creating a positive feedback loop of inflammation in the body. So now, like I mentioned, this inflammation is linked to a whole slew of different diseases in the body. The first that was kind of discovered mostly just out of coincidence with CHIP being studied in cardiovascular cohorts is CHIP is linked with a significantly increased risk of cardiovascular disease, almost on par with smoking. So that's a really significant enhanced risk of a very serious disease. Cardiovascular disease is obviously one of the top killers in Canada. It rivals really only cancer itself. But on that note, CHIP has been relatively understudied in solid cancers in comparison to a number of these other diseases I have on the slide, which is a little bit interesting to us and obviously a question that we wanted to dive into a little bit further. So CHIP is quite common in patients with solid cancer. It's present in about 20 to 30% of people with solid tumors. To put that number into context, that's about 80,000 Canadians that are diagnosed with cancer each year, which represents a huge opportunity for us to better understand the immunology of people's cancers and perhaps provide an opportunity to intervene and improve the cancer care for these people. Now, CHIP, if you remember the number I cited earlier, is actually more frequent in cancer patients versus the general population. Again, like around 20 to 30% of people with cancer versus around 10 to 20% of elderly folks. And this is due to a couple of different reasons. The first is shared risk factors between CHIP and cancer. Of course, CHIP is a kind of pre-malignant condition, cancer being a malignant condition. So there are factors like germline genetics and smoking that can increase your risk of CHIP. In addition to that, the actual treatment of cancer can predispose to CHIP, most particularly radiotherapies and cytotoxic chemotherapies. When you receive these treatments, they, of course, are doing a lot of things in the body and fighting your cancer, but they also provide an advantage to certain stem cells in the blood that carry potentially cancer-causing mutations that are found in CHIP and allows these cells to continue growing and proliferating and actually providing a risk of a secondary or therapy-related leukemia. And then finally, what I think is actually the most interesting is that CHIP might actually be increasing someone's risk of cancer. So over longitudinal cohorts and in large population studies, CHIP has been found to increase the risk of actually developing a cancer over time. And the mechanisms for this aren't exactly clear yet. They've been a little bit more studied in the context of lung cancer and liver cancer, and it's thought that the inflammation propagated by CHIP is helping spur that initial tumor growth. And once a cancer is actually diagnosed, CHIP also has some pretty important implications. So this was a seminal study out of Memorial Sloan Kettering from several years ago in 2017. This paper studied about 10 to 15,000 patients with advanced cancers. And what they found was that in having CHIP, these patients were predisposed to having poorer clinical outcomes, particularly related to their overall survival. And this was really important for a couple of reasons. The first was that most of these poor outcomes were not driven by therapy-related cancers. Most of these deaths, because they were cancer-related deaths, were related to progression of the primary tumor. And that raised questions about whether CHIP was actually interacting with tumor biology or potentially response to treatment and leading to these poor outcomes. What I should note about this data, though, and it's a confounding factor that was also rightfully pointed out by the authors, is because these patients were so advanced at their time of enrollment in the study, they had already received a number of previous lines of treatment. So these are things that include chemotherapy and radiotherapy that can cause CHIP or really bring out CHIP in someone that has a very small mutation in their stem cells. And the reason that that's really important is it introduces a key confounding factor in this data, wherein people with very advanced cancers and very aggressive cancers to begin with were receiving more aggressive treatments and aren't necessarily experiencing poorer outcomes as a result of their CHIP, but CHIP is more of a byproduct that's appearing as a consequence of their treatment for their cancer. And this is a debate that's kind of continued over the years. There's been a number of studies that have conflicted with these initial findings. The first came out not too long after And it was looking specifically at lymphoma and outcomes following hematopoietic stem cell transplantation. So stem cell transplantation is generally considered to be a curative treatment in lymphoma. So after you get it, you're essentially deemed cured and cancer-free, but of course the tumor can eventually relapse. And what this study found was that although there was an increased risk of mortality in these patients, that risk was localized to the patients that didn't have any cancer recurrence. So that's shown in the bottom over there with the dotted lines. Um, and what was found was that this excess mortality was actually driven by cardiovascular events and other kind of non-cancer related things that CHIP may be interacting with, but it didn't provide any evidence that CHIP was really interfacing with the lymphoma biology and increasing the risk of poor outcomes. Another really fascinating study was a retrospective study of a clinical trial in metastatic colorectal cancer. And what that study found, funny enough, was that mutations in DNMT3A, or that is DNMT3A mutant CHIP, was actually conferring better outcomes in this study, which is really surprising to see. And again, it really raised a lot of interesting questions about what CHIP is really doing in the tumor microenvironment, how it's interfacing with cancer treatments, and ultimately how it can play different roles in different contexts in cancer. And that brought us to, I guess, a very broad question of how can we more directly study the effects of clonal hematopoiesis in solid cancer, I guess, in isolation of a number of these confounding factors, and create a more, I guess, supporting theory of what CHIP is doing in the tumor microenvironment so we can eventually follow up on it with experimental studies. And that led us to the idea of looking for mutated clonal hematopoiesis immune cells in the tumor microenvironment. So there's been a couple studies now that have shown that it is possible to find these mutated cells in the tumor microenvironment. Again, these are mutated immune cells that are entering the tumor. They're not tumor mutations in and of themselves. And what we thought was that the infiltration of these mutant immune cells is what's contributing to inflammation in the tumor microenvironment. And this is what's ultimately shaping the tumor biology and promoting these adverse clinical outcomes that we're seeing in cancer. So to study this, we had a kind of few main goals. The first was to survey what CHIP, I guess, looks like mutationally in a cohort of solid cancer patients. Most specifically, we wanted to look at treatment-naive cancer patients. And the reason for that is it eliminates the risk of any confounding due to treatment-related effects occurring prior to the study. Once we find CHIP, of course, we want to determine what the effects of it are on the clinical outcomes of these patients. And then finally, we wanted to characterize the inflammatory associations of CHIP in the tumor microenvironment so we can understand potentially why these clinical outcomes are occurring. So to study this, we turned to two major cancer genomics cohorts that were, I guess, procured and assembled by the NIH. So these are the CPTAC cohort, which stands for Clinical Proteomics Tumor Analysis Consortium, and the TCGA cohort, or the Cancer Genome Atlas. For those of you that remember my presentation last year, I presented mainly on the TCGA data. We do have some, I guess, concerns with the quality of that data, namely in the sensitivity of the assays that were used to detect CHIP. For that reason, I'm not going to talk too much about the data here, but I can circle back to it later if anyone's curious. But we mainly focused on the CPTAC data set. And with this data, what we did was we first repurposed the genomics data, which is whole exome sequencing data. We repurposed this data to detect CHIP using existing clonal hematopoiesis detection pipelines. And then from that, what we did was we correlated CHIP status in each individual first with clinical outcomes and then also with the tumor multiomics data that was available, which includes RNA sequencing, proteomics, and phosphoproteomics as well. So to start with our overarching study objectives, let's actually find clonal hematopoiesis in these people. So to do this, like I alluded to, we're first going to start by looking for clonal hematopoiesis in the blood, because of course that's kind of the gold standard for looking for CHIP. To do this, I employed a method developed by Caitlin, a previous PhD in our lab. This is a method that is, I guess, geared towards calling or identifying CHIP in large cohorts, while also mitigating the risks of having any potential sequencing artifacts or germline variants included in with the CHIP calls, because obviously those two things are not CHIP. Instead, they're, I guess, some sort of just underlying genetic condition or something that's just not there entirely. So I applied this pipeline to detect CHIP in this cohort. We found CHIP in about 15% of individuals, or just under 300 out of our total of around 1,500. And from that, then we turn to the tumor whole exome sequencing data that was available. So we looked at the tumors of just these 300 people, and we looked for the exact same mutation that we found in the blood, representing, I guess, a movement of these mutant immune cells from the blood into the tumor. And in this case, we found that about a third of those with CHIP, just up here, were found to have what we call CHIP tumor or CH2. And what this represents is, again, the movement of these mutated immune cells into the tumor in an amount that was quantifiable by whole exome sequencing. Now, for those where we weren't able to detect clonal hematopoiesis in the tumor, we labeled those as CHPB or peripheral blood, indicating that their CHIP clone was localized to the blood and we weren't able to detect it in the tumor. Now, there were a couple different determinants of what CHIP tumor really is. We had some ideas based on just how we were defining it and looking for, enriched populations in the tumor. So the first thing we noticed was that variant allele fraction, which is, I guess, the fraction of mutated cells in the blood, or was predictive of whether these mutations were detectable in the tumor. So in the blood, a variant allele fraction roughly equates to, the percentage of VAF is equated to the percentage of reads in sequencing that carry the mutation. So for example, in CHIP, a common threshold is 10%. So you compare what's below 10%, which is generally considered to be a small clone, or something that's greater than 10%, which is considered to be a large clone. And these percentage numbers tend to double when you look at the actual number of cells that are mutated, because CHIP mutations are often heterozygous. So if 10% of mutant reads are detected, that means 20% of all of your immune cells are impacted by this CHIP mutation. And what we found was that in these cases where 20% of immune cells were affected, they were much more likely to be able to trace those mutations into their tumor sample. You can see over here, oh yes, you can't see my mouse. You can see over here that this red bar is greatly enriched, showing that these large clones are more likely to enter the tumor. What was interesting, though, is that clone size was not the only determinant of what was going on here. And you can see as I've plotted here the different variant allele fractions in the blood and tumor samples in these patients, there's a large number of CHIP tumor cases where the VAF is a lot smaller, but the CHIP clone was still able to make it into the tumor despite this, which is a little surprising to us. And the reasoning behind this was when we eventually turned to the RNA or transcriptome of these tumor samples, we found that the samples with CHIP tumor tended to have greater immune infiltrates overall, indicating that there was a greater number or proportion of immune cells in this tumor. So the combination of more or a greater fraction of your immune cells being mutated and more immune cells getting into the tumor, those were kind of the two main things that were predicting the presence of what we've called CHIP tumor. So now that we've identified CHIP tumor or CHIP and CHIP tumor in these samples, we wanted to see what the clinical implications of this were. So first, we looked, I guess, more traditionally, like those studies that I've shown you previously, at just CHIP as an overall phenomenon, looking only at the blood, whether or not we detected CHIP, what were the clinical outcomes. And what we found was that CHIP actually didn't really do all that much. You can see the hazard ratio is just hovering around 1. It's completely non-significant. There's not really any trend towards a specific effect one way or the other, which is suggesting that CHIP overall isn't super meaningful in this context of primary treatment-naive cancers. What was interesting, though, is the moment we split things apart by CHIP tumor and CHIP in the peripheral blood, that's when the effect really started to take hold. So you can see this purple line over here is what we've called CHIP tumor. So these are the cases that we thought in advance were going to be affected more prominently, and that was the case here. So these cases with CHIP tumor were experiencing significantly poorer survival outcomes, whereas when CHIP was only in the peripheral blood, or CHIP-PB, this green line over here, you can see that the outcomes were almost identical to those without any CHIP at all. So what this is suggesting is there seems to be a bit more of a dose dependency with CHIP, where there needs to be a sizable proportion of mutant cells inside the tumor in order to have some sort of effect on clinical outcomes. And we didn't have definitive cause of death information for a lot of these patients, but what we did have is whether or not they were considered to be tumor-free at their last known point of follow-up. So it's not exactly a cause of death, but it does suggest whether or not a patient was considered to be, I guess, cured or relapse-free from their cancer, depending on the different type of cancer they had. And what we found here is, as you can see right in the middle with CHIP tumor, it was the only condition where patients were significantly less likely to be tumor-free at their last known point of follow-up, which is again suggesting that their cancers are sticking around, they're harder to treat and tend to be more aggressive. And the clinicians were having trouble, I guess, treating and I guess completely removing these cancers. So then finally, now that we've seen that there's some sort of clinical effect related to CHIP tumor, we wanted to dive into the molecular data related to these tumors to see if we could identify any associations with CHIP that could point towards potential mechanisms that might be driving this change. So the first thing we did was we looked at differential gene expression analysis in the tumor samples of these patients. So we took the tumors of those that were considered to have CHIP or CHIP tumor, and we compared those to the tumors of people who were not considered to have CHIP at all. And what we found was there were a number of significantly differentially expressed genes. These are genes, I understand it's a little bit hard to read on the slide, but these are a number of genes that are related to a number of different inflammatory pathways, including interleukin-1, C-reactive protein, which is more of a general marker of inflammation, and a number of other immune-related genes. I couldn't really list all of them here, but that's just a quick summary. And what we also noticed was that of the genes that were differentially expressed in the context of CHIP tumor and in CHIP as a whole, the magnitude of change was far, far greater in the context of CHIP tumor that you can see over here. And what that's suggesting is many of the signals that we're seeing when we look at CHIP overall are mainly being driven by this subset, or about a third of CHIP cases where the mutated cells are entering into the tumor microenvironment. Now, circling back a little bit, this is where we wanted to revisit some of that TCGA data that I talked about. Although that data was limited, there were some inflammatory signals that we were able to tease out. And what we wanted to do here was compare the signal that we were seeing in both cohorts to see if the signal we were having in the CBTAC set was a little bit more of an artifact, or if it was something that could be reproduced in more than one cohort. So what we did here is we looked at the overlap between differentially expressed genes in both cohorts, and we looked for pathways that were enriched in these overlapping genes. And as you can see here, there were a number of important and relevant pathways that were showing up, including antigen presentation through major histocompatibility complex, and then also interleukin-6 production. And interleukin-6 was really one of the most exciting things we saw, because we know that IL-6 is one of the most prominently upregulated signaling pathways in the context of CHIP, and it suggests that it might be one of the main axes that is driving some of these survival effects that we're seeing. Now, at the pathway level, what we wanted to do is look at the differential expression of gene sets at an entire pathway. So we looked at a number of different pathways that were related to inflammation and inflammatory signaling. And what we found was that first, looking at CHIP as a whole, which is this bar really just on the far left, far left, there really wasn't any significant effect that was related to CHIP. So given all those previous studies, and even given our survival findings, there wasn't much of a clinical change that was associated with having CH, and there's also not much of a change in gene expression either. What was really interesting, though, is again, once we parsed the data by whether or not the mutant cells were in their tumor, that's where we saw a really interesting effect. So what we see is on that second column is CHPB, where the mutations or the mutated cells weren't able to enter. And in this case, there seems to be a suppression of a lot of these immune and inflammatory related pathways. On the other hand, when we look at CH tumor, there's this kind of opposing effect that gets canceled out when we look at CHIP as a whole, and that's where a lot of this inflammatory signaling tends to be enriched. Now, this is probably a good point to note that this is not necessarily showing that CHIP is causing a lot of the signal that we're seeing, but it is suggesting that CHIP is associated with these signals. So it's still important to tease out whether these cancers, or whether more inflamed cancers are more predisposed to recruiting CHIP into their environment, or CHIP is helping to actually shape the environment to create a more inflamed cancer. Those are still some things we need to follow up on and tease out. But a common question that we get related to this data is, how does this compare to the effects of age on gene expression in the tumor? And what we found was that these inflammatory changes are distinct from aging. So at first, we didn't want to directly control or adjust for age in those analyses I showed previously. And the reason for that is because age and the presence of CHIP are so closely linked, we didn't want to interfere with any signal we saw. So instead, what we did was we looked particularly in the individuals without any CHIP at all. And we asked what was the effect of aging in these people. And what we found was that, across a number of inflammatory pathways, the expression of inflammatory pathways in these cancers was dampened or suppressed. And what you'll notice is that this signal that we see related to age in individuals without CHIP is actually quite similar to what we see in the individuals with CHPB, or again, CH in the peripheral blood, not in the cancer. And what that's telling us is that these people with CH, who on average are older than your average patient with cancer, but without any clonal hematopoiesis in their microenvironment, are experiencing generally age-related effects, and it's not really an effect that's specific to the CHIP that they're carrying. Whereas in the case of CHIP tumor, when these mutant cells are getting into the cancer, that's where we start to see things changing and ultimately, again, linking back potentially with the clinical outcomes. And while we were on this page, thinking about how aging might be influencing the effect of CHIP in cancer, we came across this study that came out last year that was studying mainly lung cancer, but in a number of other cancers as well, a phenomenon called age-related emergency myelopoiesis. So what this, I guess, what this mouthful really is when a cancer is present, there seems to be this IL-1 mediated signaling axis that drives enhanced production of myeloid cells, particularly slightly immature progenitor-like myeloid cells that are able to get into the tumor and ultimately interfere with the anti-cancer immune response and drive poor outcomes. What was really interesting is what we noticed in the study is they found that this effect was associated with an aging, or what they call an aging-related decline in DNMT3A function, which was pretty interesting to hear because, of course, we know that there's also an aging-related decline in DNMT3A function with CHIP when we lose the function of DNMT3A with a mutation. Now, of course, we wanted to expand this idea a little bit further, so we also looked at other drivers of CHIP, like TET2 as well, which we know is able to create somewhat convergent phenotypes compared to the loss of DNMT3A. And what we found was that CH is really just intrinsically linked with this IL-1 alpha expression in the tumor. So first, I'll show you, this is just the same data that was on the volcano plot a few slides prior that shows that the expression of IL-1 alpha on its own was significantly enriched in the context of CHIP tumor, but not in the context of CH in the peripheral blood. Now, what we did was we looked at not only IL-1 alpha expression, but also the expression of an IL-1 alpha gene expression program, which is a number of genes that are co-expressed with IL-1 alpha, and we looked to see how the expression of these pathways change in response to CHIP and in response to aging. So, of course, we see with CHIP tumor that these programs are enriched. And in the context of age, we were actually able to repeat the findings of this study. So looking across all patients with increasing age, so that's shown up here, and then over here in the middle down here, that with increasing age, agnostic to a patient's CHIP status, IL-1 alpha expression is enriched, which is in line with that study's previous findings. What was really interesting, though, is that when we looked at the effects of increasing age solely in the individuals without any CHIP at all, which is over here and down here, this effect entirely disappeared, which is suggesting a lot of this, I guess, age-related quote-unquote IL-1 alpha production might be mediated by clonal hematopoiesis instead. And a really important implication of this is the previous study showed that this IL-1 alpha mediated signaling axis is targetable by anakinra, which is a clinically available antibody. So it highlights one possible mechanism that we might use to, I guess, target this clinical association of CHIP on patient survival. And on the topic of clinical associations and treatment, we also took an exploratory look at what CHIP might be doing to immune checkpoint inhibitors and the response to those types of treatments. So we know that these treatments do not necessarily work in everybody, but there are some predictors of survival and improved response to these anti-PD-1 and other immune checkpoint inhibitors. So we took gene sets that were, so these are collections of genes that predict a more productive response to these treatments, and we tested whether they were enriched in the context of CHIP. And again, what we saw is that in CHIP tumor specifically, but not in CHIP in the peripheral blood, these pathways were enriched, suggesting that CHIP might play a role in improving immune checkpoint inhibitor response. And this data on its own isn't super convincing. I know it's a little bit exploratory, but we do have some data produced in collaboration with Dr. Vanner and some of our other colleagues that's showing that TET2 mutant clonal hematopoiesis might actually be promoting or enhancing the response to immune checkpoint blockade. So I can talk about that a little bit more later if anyone's interested. So now another major question that we had with this work was, how is CHIP interacting with different types of tumors and in different tumor microenvironments that might be mediated by different parts of the body, for example, in the brain or the colon or the breast? Is the inflammatory signaling associated with CHIP leading to different changes in the tumor microenvironment? And I don't really have time to go through all of this data and all of these different cancers that were included in CPTAC. But I want to specifically draw your attention to the data we have for glioblastoma, which is just abbreviated as GBM here, where we had a really strong signal related to clonal hematopoiesis in the tumor microenvironment, where there was a lot of inflammation in these cancers. And this excited us for a couple reasons. The main one is that myeloid inflammation is really important in glioblastoma and the development of these cancers. So this is a vast oversimplification on the slide, but I've included it here just to set the context a little bit. Myeloid cells are really, really abundant in the glioblastoma microenvironment, upwards of 30% of all cells in the tumor mass. That includes the tumor cells as well. And they play an important role in interfacing with these tumor cells and shaping their phenotypes and ultimately co-evolving in the tumor microenvironment to shape the type of tumor that between different types of macrophages in the tumor microenvironment. So the first, there are microglial-derived macrophages, and these are macrophages that come from the resident microglia in the brain. So these are cells that are not produced by the bone marrow and would not be carrying any CHIP mutations. However, as glioblastomas tend to progress and take on a more mesenchymal or stem-like phenotype, there tends to be more and more infiltration of monocytes or bone marrow-derived cells into the tumor microenvironment. And these monocytes are the ones that could potentially be carrying CHIP mutations. And what happens is, as more and more monocytes enter into the glioblastoma microenvironment, they tend to upset the balance of these tumors, and they actually lead to enhanced aggression and recurrence risk of these cancers as well. So we wanted to dive into this signal that we were seeing and see how CHIP might actually be playing a role in, I guess, shaping the glioblastoma microenvironment and potentially changing the course of the tumor development. So the first thing we did was we looked at some of these different subtypes of glioblastoma. And what we found, most notably at least, was that in the context of CHIP tumor, which is that bar, again, right on the right, there was an enrichment of mesenchymal subtype tumors. So these, of the patients with CHIP tumor, they were much more likely to have this mesenchymal and aggressive and monocyte-rich cancer. Again, it's not clear whether CHIP is causing these cancers outright or shaping the tumor's development into that mesenchymal type cancer, or whether mesenchymal cancers are good at recruiting these CHIP cells. But it was really interesting for us to see that how closely the mesenchymal subtype and CHIP were aligned. What was also interesting is in the clinical outcomes of just glioblastoma patients in particular, CH tumor was again associated with poor outcomes. Now, this was a bit of an exploratory look. The sample size was a little bit smaller, but at least in a univariate model, we saw a significant reduction in overall survival in the context of CH tumor, which is again suggesting it's associated with a more aggressive tumor phenotype. So now to try to tease into this a little bit more, what we did was we looked at CH tumor in the context of mesenchymal glioblastoma specifically. So we wanted to create a bit more of an apples to apples comparison, looking only at these mesenchymal tumors and comparing those that had CHIP in the tumor microenvironment to those without. And in this case, these are the results of a gene set enrichment analysis looking at a number of these pathways. And what we found was that shown in red is enrichment of a number of pathways. So these are pathways that include immune regulation and immune cell differentiation, but also pathways that are involved in extracellular matrix organization. So these are things that are important for cancer progression and growth. So these are processes that are not only highly important in glioblastoma and cancer in general, but these are processes that are exacerbated in the context of CHIP, which is again suggesting that CHIP is playing a more important role in shaping how these cancers develop. And then finally, what we've also shown is that at the level of the kinases or phosphoproteomic signaling in these samples, there was an enrichment of a number of different signaling pathways that are related to tumor growth and development. Peter, sorry, I realize you are having trouble seeing that font over there.
No,
Okay, but it is related to, there's a number of pathways related to angiogenesis and immune migration that are, again, suggesting that these tumors are more aggressive when CHIP is present. So of course, this kind of brings me to the end of my talk on some of the data that we have already, but it does bring up a couple important conclusions and future questions. And the context of these is pretty important for where we want to take our work in the future. So our first kind of major conclusion is that CHIP in the tumor microenvironment is common in cancer, and it's able to effectively stratify CHIP to identify those who are at risk of adverse outcomes. So now from this, what will be really important is determining what variant allele fraction threshold in the tumor tissue is defining clinical relevance. So what we've done in this study is we've looked at CHIP as a binary, looking at either the presence or absence of CHIP in the tumor, and that is a function of the data that was available to us with the whole exome sequencing. What we plan to do in the future is use more targeted and sensitive approaches to detect CHIP. And the reason we want to do that is to detect truly all of the CHIP clones that are present in these samples, not just ones that are large enough for us to detect. And that will enable us to, I guess, determine what extent of mutant cell infiltration is enough to lead to some of these outcomes that we're seeing here. Another important question that's a little bit outside of what I've been currently working on is how can quantifying the infiltration of these mutant cells provide value in other age-related disease contexts. I'm studying solid tumors, obviously, but a lot of my colleagues in other fields are studying the role of CHIP in cardiovascular disease and kidney disease and liver disease. So it'll be important to see how infiltration of CHIP into the tumor microenvironment will play a role in how we're understanding the role of CHIP in these diseases and potentially lead to any kind of new discoveries. Another kind of main takeaway from this work is that the infiltration of these CH mutant leukocytes into the tumor is altering immune profiles and tumor biology. Of course, a really major point of follow-up for this work will be determining what are the signaling mechanisms that underlie crosstalk between CHIP mutant cells in the tumor within the tumor microenvironment. Of course, we have some hints and signals from what we've seen in our data based on our gene set enrichment analyses and differentially expressed genes, but it'll be important to model this and determine this in a more, I guess, exact manner so that we can one day target these and potentially leverage them during cancer treatments. Another kind of wider question is how does CH actually shape evolutionary pressures during tumorigenesis? So we know that CHIP can potentially increase the risk of a cancer developing, which is suggesting that CHIP and its inflammation is important for allowing a tumor to grow. But another question is how this inflammation can shape the type of tumor that grows. So is the inflammation that's present selecting for particular drivers or cancer clones or subclones during the process of tumorigenesis that's shaping the eventual type of tumor that develops, potentially to be more aggressive or responsive to a particular type of therapy? Another kind of offshoot of that question is, are some tumors actually better at recruiting CH mutant cells than others? Well, what we've seen with my data is that clonal hematopoiesis in the tumor microenvironment is associated with poor outcomes, but it's still an outstanding question as to whether these more advanced and aggressive cancers are better able to recruit the immune cells, or whether the recruited immune cells are actually shaping a more advanced cancer, or perhaps a combination of the both, where there's some sort of coevolution between both factors that's leading to the poor outcomes that we're seeing. So now to follow up on this, of course, I still have a whole degree ahead of me. So there's a couple more things that we want to do to study this. The first is to characterize CHIP in the glioblastoma microenvironment using single cell technologies. So to do this, what we've done is partnered up with some researchers at the University of Calgary through the Marathon of Hope Cancer Centers Network. And what they've done is they've collected a number of glioblastoma samples with matched blood from patients with this cancer. And what we plan to do is first to detect CHIP in these samples using targeted sequencing and whole genome sequencing, which will allow us to find CHIP in the peripheral blood and in the tumor microenvironment. And then from that, what we want to do is two kind of main technologies. The first is applying more of a classical single cell approach, or in this case because it's brain cell single nuclear RNA sequencing, and that will enable us to determine where these inflammatory changes are coming from. Of course, we've seen a lot of changes in cytokines at the bulk level in these tumors, but it's important to understand where these signals are coming from. Are these cytokines being released by the tumor cells? Are they being released by the macrophages specifically? So that'll be something that's really important to tease out in order to get a better understanding of what's going on. But importantly, on top of regular single cell sequencing, we want to layer a technology called genotyping of transcriptomes, or GOT, over that. And I can talk about that technology a little bit in the discussion period, but essentially what it's doing is overlaying single cell genotyping with the transcriptomic profiles. And it allows you to merge, I guess, both types of data together and compare the single cell gene expression profiles of cells with CHIP mutations versus those without. So that will allow us to compare within a single tumor how the mutant CHIP cells are differing from the wild type immune cells in the tumor. And then finally, what we also plan on looking at is spatial analysis in these tumors. So that for that, we plan on using 10x Visium technology, where there's essentially a tissue slide, and after you place a tumor tissue on, there's spatial capture regions. So rather than coding single cells, as in a classical, or I guess 10x chromium experiment, what's happening is the cells are being, or the gene expression is being tagged by the specific location on the slide, which roughly correlates to single cells or groups of cells, but not exactly. And of course, from all this, again, we're looking at associations between CHIP and cancer in the tumor microenvironment, but we want to move even further ultimately to determine mechanisms behind a lot of these changes. And that's where experimental modeling of CHIP and glioblastoma will come in. And here I want to thank Daryl Chan over in the Vanner Lab for helping to get this model established and working to get everything running smoothly here. But essentially what we want to model is clonal hematopoiesis in glioblastoma using a mouse. So this model starts with CRISPR-edited human hematopoietic stem cells. These right now we're trying TET2 knockout human HSCs, but there's also potential to try other genetic drivers of CHIP, such as DNMT3A. And after CRISPR editing or CRISPR knockout of these genes in hematopoietic stem cells, what we're able to do is inject these HSCs into an immunodeficient mice. So this is a mouse without any immune system of its own. So it becomes reconstituted with the human immune system. And then once this human immune system is present, we then plan on injecting glioblastoma patient-derived xenograft cells into the mouse's skulls. And then that will allow for, I guess, a human glioblastoma to develop in the mouse and interact with human immune cells. And that will allow us to better understand how the mutations in clonal hematopoiesis can ultimately shape the development of these cancers. So with that, that kind of brings me to my overarching conclusion, which is that CHIP is something important, something that deserves a little bit more consideration in solid cancers in order to discover how and why our tumors are developing and why our immune systems are responding differently to different cancers. And we think that by, I guess, flipping the script a little bit in precision oncology and considering a patient's immune genetics in addition to their tumor genetics can be really valuable and provide a lot of hope for the future of immunotherapy of cancer. So with that, I just want to quickly thank you all, of course, for listening to my talk today. Thank you to my supervisors, Mike and Rob, who's online right now. You guys have been super supportive throughout this whole process. My lab mates, you guys are lovely. Peter, my supervisor committee, thank you for guiding me here. Thank you to all of our collaborators. And of course, now, like Peter mentioned, this work is now published. So if you do want to read it at all, go nuts. But yeah, that's about it for me. And I'm happy to take any questions you guys might have.
Thanks for the last questions. Who would like to do it? I see a hand.
Wait for the students.
Oh, you can start.
I don't think you know how it works here.
Go for it. Great targets.
Thank you.
And like you almost finished your PhD and it's like a double PhD. So much more you can do with so many questions. And a very exciting project. So it's kind of counterintuitive, right? Like poor response with more CHIP, because if more infiltration is there in solid tumors, especially in the context of chemotherapy or radiation or immune checkpoint, there is this concept of hard a more infiltrated immune environment, pre-treated immune environment, and a good response. So where does this fit in? The chip mutant immune cells that are recruited are these pre-existing, exhausted, they're non-responsive, they're just coming because of this chemokine filter, or what's happening there? What's the mechanistic basis?
Okay, yeah, thank you. That's a really great question. Just to repeat it for online,
Yeah.
is with all of these immune cells that are infiltrating into the tumor microenvironment, some of them, of course, carrying chip mutations, our findings a little bit counterintuitive based on our understanding of tumors that are necessarily hot or cold based on the types of immune cells that are getting in. That's a really great question and something I wish I had a little bit of a better answer to. We don't know too much about the specific types of immune cells that are getting in. What we do know is that the mutant cells are typically in the myeloid lineage as opposed to the lymphoid lineage. What's a bit of an aside, but what is interesting is that when TET2 and DNMT3A are lost in lymphocytes, like in CAR T cells, the cells actually become more effective at fighting cancer. But typically in, I guess, a natural course of clonal hematopoiesis, most of the mutations are carried in the myeloid cells. And what they're necessarily doing in the tumor, we don't quite know yet. We would presume that there's quite a bit of crosstalk between the myeloid lineage, or those that are primarily affected by the mutations, and the lymphoid lineage. And that would, of course, interfere with how the tumors are actually responding to their treatment. What was actually really interesting is we looked at survival outcomes related to just immune infiltration on its own. Sorry, I forgot to mention that earlier in my talk. The findings in survival related to CH tumor were actually already adjusted for immune infiltration, and immune infiltration on its own was not able to predict survival, at least in this cohort. I know it does vary a little bit from cancer to cancer, but at least overall, that was not the case. But anyways, coming back to what I was saying, we want to know more. And that's kind of where a lot of our next steps are coming in to see what exactly these mutated cells are doing in the tumor microenvironment. Are they solely releasing inflammatory cytokines? Are they directly interfacing with cancer cells? Is it more because the myeloid cells are interacting with the stroma to recruit more cells, or interacting with the lymphoid cells? Could be all of those things, could be some of them. And of course, the specific mechanisms are still unknown, but something we definitely want to dive into a little bit.
With aging, as you know, many of these immune cells populate peripheral sites. Right?
Mm-hmm.
So, in a non-cancer scenario, you think that mutated cells are infiltrating an aged peripheral organ, like let's say some organs are heavily infiltrated, some others are lung is right, and mucosal sites. So they acquire a residency kind of thing, and when cancer sets in, how does that? Have you looked at any data that you have pre and post kind of scenario in treatment? From the GTA's database or something like that?
Mm-hmm. Yeah, no, that's another really good question. No, it's all good. To repeat that for online, Dr. Cody was asking about chip that's infiltrating just other types of organs and tissues in the body in the absence of cancer, and how that might play a role in, or once cancer develops. And that's a question I've been wondering about for a while. We're really curious about how chip might actually be, I guess, shaping or orchestrating, I guess, a precancerous or pre-malignant niche. What we know, well, I would, to briefly answer your question, I would think that chip mutated cells are getting anywhere that your peripheral blood immune cells are going. So just from like probability, if half of your immune cells are affected by these mutations, if immune cells are getting in, if there's more than 2 immune cells getting into a specific tissue, I would put my money on the fact that chip mutant cells would also be there. In the context of what they're doing to, I guess, condition the microenvironment for chip, that is where I think the longitudinal studies would be really interesting. I unfortunately don't have access to pre and post data. That if you do know any, please let me know, because I'd love to take a look. But that's where the longitudinal studies can be really helpful. So looking at things like the UK Biobank or All of Us, and these are population studies of hundreds of thousands of healthy people, and they have data following these people's health in the long term. And what we've seen in studies like this, I can actually flip to, I've got an extra slide about this. Just give me 1 second. What we see in cases like this, is that CHIP can actually increase the incident risk of solid cancer, which is making us think that the presence of CHIP in the pre-malignant environment, for example, the lungs, these mutant cells are again releasing inflammatory factors and conditioning the surrounding cells to create an environment that promotes the initial growth of a tumor. And you can see this is data from another study in the UK Biobank. Of course, hematologic cancers are lighting up left and right because CHIP is, I guess, directly evolving into a hematologic cancer, but a number of solid cancers are also enriched, both overall in that column kind of in the middle on the left, but then also in a number of other cancers, including lung cancer, which is really interesting. So CHIP has been associated with an increased risk of lung cancer. And what we've actually done is we looked at this data in the context of air pollution. So there was a study from Charlie Swanton's group a couple of years ago about how air pollution can create a lot of inflammation in the lung environment, and that promotes the growth of these initial lung clones. And what we found is that CHIP was actually mediating this relationship. So it was, or I guess the inflammation induced by air pollution alone was not enough to induce lung cancer, or at least increase the risk of having lung cancer. But when you had both the high exposure to air pollution and CHIP, there was this synergistic relationship where I guess the inflammation was kind of brought out more in a more direct or severe way, and that was able to increase someone's risk of developing lung cancer. But yeah, so lots more to do, but yes. Mm-hmm.
I noticed you had COPD on your introductory slide. Did I see that
Yes.
right? So what's known in COPD is there a correlation also?
Yeah, there's a correlation between CHIP and just about like every age-related disease you can name. I only listed a couple on the slide just because there's only so much room on that one guy. But if it hasn't been studied or published already, there's probably someone looking at it. Because it's a fairly easy thing to study and get a paper on. It's just like, oh, CHIP is linked with this phenotype, voila. In COPD, there is a little bit more information about mechanistically what's going on. So again, macrophages and hyperinflammatory macrophages can be releasing a lot of soluble factors into the lungs, which helps activate some of the cells in the surrounding environment. I'm not super familiar about the exact mechanisms that are going on in COPD. The other thing is there might be some interactions with smoking and how that develops. So smoking particularly predisposes to CHIP with mutations in a gene called ASXL1. So it's the third most common cause of CHIP after DNMT3A and TET2. And so with smoking and ASXL1, you kind of get this double threat in the lungs where there's inflammation coming from the CHIP mutant cells itself. There's also a lot of damage coming from smoking. And those, I would presume, are really, I guess, wreaking havoc on the lungs and breaking down a lot of the lung tissue and leading to lung disease. But yeah, essentially any disease, lungs, heart, liver, kidneys, you name it, someone's probably looking at it.
I'm guessing people have started to do some single cell RNA sequencing data to do one of the things that you, I guess, alluding to there is try to parse it out and find out what's different about the CHIP cells compared to.
Mm-hmm.
So what's known? What have people seen so far doing that? Or
yeah, so a lot of things have been done already. There's been studies at the level of the stem cell and like the HSCs, which I alluded to in my introduction, and then there's been studies in the PBMCs, or I guess peripheral blood cells that are actually circulating in the body. At the level of the HSCs, mutations like DNMT3 and TET2 are the most commonly studied. And what we found, or not we, what people have found is that at the stem cell level, these mutations confer a resistance to inflammation. So these cells are able to continue growing and dividing in the context of inflammation, whereas your healthy cells kind of regress and hide away. What's really cool, though, is that the mutant cells are actually communicating with the wild type cells. So the healthy, unmutated stem cells in someone with CHIP are more resistant to inflammation than someone without CHIP at all, which is really interesting.
but I was thinking of more.
Yeah, and then so in the peripheral
blood,
blood,
can you actually see the difference in phenotypes of these cells, and does it correlate with the expectation of the more inflamed cell type?
yes. So they don't cluster exactly the way you do, like between CHIP and no CHIP necessarily. They'll still cluster based on their wider cell type, like monocytes or neutrophils, et cetera, et cetera. But in people that have layered, like for example, genotyping of transcriptomes over single cell data, The mutated cells, again, tend to take on this more inflammatory phenotype. They tend to secrete a lot more pro-inflammatory cytokines. Their phagocytosis is a little bit different than normal. In the context of neutrophils, there's disruptions in netosis, so they're not really able to release those neutrophil extracellular traps like they normally would. And again, actually, in not just at the HSC level, but at the peripheral blood level, even the healthy cells in people with CHIP tend to be more inflamed than the healthy cells in someone without CHIP at all. So that, again, there's still that kind of systemic crosstalk that arises from those.
Question about the association between CHIP and cancer incidence. I don't think you've shown several studies suggesting that there's a higher incidence of CHIP and cancer. And how do you know? Are you proposing that that's a sort of an inflammatory trade-off of the CHIP that's producing the cancer, or are these individuals who've got inherent problems with genomic integrity, genomic maintenance, and therefore they come up with these clones of cells, CHIP and cancers?
It's a good point. I think it's a little bit of both. So in the context of like lung cancer, for example, smoking obviously plays a big role in this. That's why we looked at people who haven't smoked at all. Because of course, smoking increases the risk of developing CHIP, but also independently the risk of developing lung cancer. Same thing with germline genetics. So there are some oncogenic, like there was a study, I don't think it's published yet, but it's looking at a number of different germline mutations that are known to predispose to cancer. So these are mutations in a lot of genes that are involved in replication fidelity in the DNA damage response, genes like CHEK2 and ATM. And mutations in those genes obviously predispose to cancer, but they also independently predispose to the development of CHIP. So that's kind of one way that CHIP is more common in cancer patients. The other side of it, of course, is this more inflammatory axis that we think is supported by this data here, but also some other studies. Like, for example, just to flip to the previous slide, this study in patients with liver disease. This specific type of liver cancer often arises out of a liver that's already quite damaged and inflamed. And what those authors think is happening, and what I tend to agree with, is that the mutated cells releasing producing inflammatory signals and acting in a hyperinflammatory manner in the liver tissue is helping promote that growth, or that initial spurt of cancer growth that leads to a tumor. So I think it's a little bit of both. Parsing that will require just a little bit more information about the nature of CHIP. Once we know a little bit more about the germline genetics, we can control for that a little bit better. But for now, that's kind of the best we can do. Yeah. Mike?
Quick follow-up related to that. What about, recall Marco and CANTOS at all? That study?
Yes.
Lung and lung cancer?
Yes, actually, that's a good point as well. So CANTOS was a cardiovascular study. It was a population-based cardiovascular study where patients received canakinumab, which is an IL-1 blocker, or specifically IL-1 beta blocker, which is one of the main cytokines that's enriched in the context of CHIP. And following these patients longitudinally, they found that cancer risk was actually decreased, specifically in the individuals with CHIP, which is a really surprising finding. Of course, it's retrospective and there's more study that needs to be done related to that. But just the fact that giving someone some sort of inflammatory blockade can limit the risk of cancer, specifically in those with CHIP, I think is pretty powerful, and something that warrants a little bit more study.
So I'm curious about the glioblastoma model that you described near the end. So that I can see the allure of doing this with human glioblastoma. It seems like a very complicated experiment.
It is. It is.
Compared with the alternatives, which
Yeah.
you probably have.
Yeah.
Ask. Maybe you could.
Yeah. So I will preface with that model I presented isn't quite working the way we'd hoped. The mice tend to get this, especially the TET2 knockout mice, tend to get this hyperinflammatory syndrome, and it obviously interferes with a lot of the experiments. The mice really aren't that healthy, so it's not working all that well. It's a limitation of the model.
So you've tried it with just your mice.
We haven't yet, so we are looking into some alternatives to model that. These are mice in Toronto. So we're just in the stages of getting the model going.
Okay.
Yeah. So
but you're talking about essentially trying to humanize a
yes.
humanized model, so that you can grow human glioblastoma cells in that mouse and have a human chip or non-chip hematopoietic.
That was the
system.
goal,
But
yeah.
I was thinking you have mice, black 6 mice that you
Yeah.
can do the same sort of experiment, assuming you have a decent engraftable glioblastoma. Do you have?
Mhm.
Engineering. So we're exploring that. Like,
Yeah.
Rob had this underway already, looking to partner with him, so. Mark was just mentioning that, but then there's one other.
Yeah,
possibility.
the other possibility we haven't
Yeah.
really teased apart too much is the potential of using organoids rather than mouse, just
Co-culture.
or mice, just to and like co-culturing the mutant cells with like a glioblastoma organoid instead. And that would obviously give us a little bit more flexibility. It's a little bit easier to do.
Yeah.
So yeah, there's a couple options. Things are still a little bit up in the air. We're getting things going at this point, but
Yeah, it's doing the gene editing right at the outset is not gonna be trivial.
not that
But
easy.
you have the mice.
Yeah.
with the editing.
Yeah.
Yeah.
Other questions for Marco? Any questions online?
I think there's something in the chat.
See one?
Oh no,
she's
just someone.
That's. Congratulations. Great topic. Excellent work and presentation.
All right.
If not, please join me.